Okay, welcome to Level 1 Science Online with Henny van Vieren.
Hey Year 11s, welcome back to another lesson on genetic variation and change. We're just going to continue where we left off last time with how new alleles are formed through mutations, and now we are moving on to those sources of variation. So here we go. Last time we looked at all of these and we saw that genetic variation happens when there's a permanent change in the base sequence of a gene which is contained on the DNA. If this permanent change in the gene on the base sequence of the DNA is contained within the gametes, in other words the egg and sperm, then it can be passed on to the offspring. However, if that change is in the somatic cells and not the gametic cells, so soma for the body, then it is going to be a local mutation and only that person will be affected by the phenotype. So in this case, that jaguar, or well, it's a leopard I think, has had a mutation and is now called a black panther. And this black panther now is a nocturnal animal, lives in a very dark forest and is very difficult to find, so really good camouflage in comparison to the normal panther next to it. So, and because of that, there was natural selection for it. Natural selection means that my phenotype gives me a slight advantage over somebody else because I am a better fit for my specific environment. And because of that, I have a higher reproductive success, and because I have a higher reproductive success, I will have basically more babies that come and have my specific phenotype because I'm passing on this new allele. Okay, so I'll make sure that when if a mutation occurs and the gamete and these gametes have been used in fertilization, then they have a possibility of being passed on to the offspring. So when we look at different sources of variation, you have asexual reproduction when we look at reproduction itself, and we have sexual reproduction. Now, asexual reproduction, there is no variation in that. Okay, that is when you take a cutting from a plant and you basically stimulate it to grow roots. So if you ever walked with your grandmother, my mother for instance, she would walk past a lovely plant and say to the owner, "Oh, please, can I take a cutting for my home?" And then at home she would put it into a glass of water, and eventually it will sprout some roots, and then you plant that, and there you go. It's exactly the same DNA as the original plant, so there's no variation happening there, and that is cloning. Okay, you can do that today if you have, for instance, have you ever seen strawberries when they put out those little pups, I think they call them as well, but it's like these little growths, and where it touches the ground it forms its own roots again, and you can take that and replant it and you will have another strawberry plant with exactly the same DNA. And when you saw in the video I had you guys watch on the bananas, where the bananas are in real trouble because they are all clones of the original Cavendish banana. So now we have the banana rust, which is coming and killing all the bananas because they all have exactly the same DNA. Isn't that weird to think about, that every time that you eat a banana you are eating exactly the same DNA? It's strange. There's something strange about it. Now, this picture over here, I used to, as you know, I did some research in the forestry industry, and this is a plantation of all clones. So all these trees that you see have exactly the same DNA. So they will share very much the same characteristics depending on the environmental conditions on them. And this is very favorable for somebody who runs a huge factory like a paper mill, because then they know what wood they can expect and what characteristics that wood would have, and they can fine-tune their process really, really well. Yeah, so this is an example of when they use it to their advantage. Now, unfortunately, some mutations can be harmful. These little spider lambs, are they called? They are most likely something like a Siamese twin that didn't separate properly. but you can see that is obviously a harmful mutation there. And this is another harmful mutation of a little kitten that was born with only one eye, and you can see there's deformities in the mouth and nose as well. So, but they will most likely die before reproductive age, even though the mutated gene came through the gametes. So in this case, it will only be passed on one generation and then nothing from there. Here's some other mutations that you might not have seen before, but this is a specific human syndrome, which is a hereditary syndrome, where the children, and this boy is about 12 years old in this photo, and he's got it, a syndrome where your cells age exceptionally fast. So his body cells are the same as an 80-year-old person's, but he's only been living for 12 years, which is really, really sad. And they don't grow very old. And then over here, these are they call themselves the Mexican wolf family. They have been born with a mutation where their faces are covered in hair. And some of them have gone on to marry people without hair, and their children now have slightly less hair. But because this is not an adaptive advantage, it will not obviously take root in society. However, if you think about it, let's say there's a sudden ice age and everybody's noses freezes off except these guys because they have hair to protect them, then that would be an adaptive advantage and that would be natural selection. But at the moment there's no selection pressure for it. So we talked about mitosis before, and like mitosis contains this big T, and it gives you two identical diploid cells. A diploid cell is got the whole 23 pairs of chromosomes, not just 23. They have 23 pairs, in other words 46. However, in the production of egg and sperm, we go through meiosis, and it contains that E, and the E is going to remind you of egg and sperm, and this is where we want to halve the genetic material. And when we halve them, it's called haploid, and we get 4 haploid cells at the end of meiosis. Okay, so important. Egg and sperm are formed through meiosis, and it is basically we halve them, because after you halve them, the egg and the sperm is going to come together in fertilization, and if they both have 46, you're going to have a real problem. That fertilized zygote will have 96 chromosomes, and that will not survive. So you need to have half. Half is going to come from the mom, half is going to come from the dad. They come together and you have a new zygote, which is your new individual. And this only happens in the ovaries and the testicles, and only for egg and sperm. Okay, so mitosis, 2 diploid cells. Meiosis is 4 haploid gametes are formed. Your 2 identical diploid cells over here in mitosis. Think about it, if you have a cut in your arm, you need the skin cell there to grow. That's why it needs to be identical to the one that is next to it. You don't suddenly want, I don't know, liver cells to grow in your cut. That'll be weird. Okay, and this is a short video on DNA replication.
To make perfect copies of our DNA, an enzyme first unwinds the 2 strands of the molecule by separating its base pairs. Another enzyme then attaches free nucleotides to the exposed sites. The existing strand determines the composition of the new strand. According to the base pairing rules, adenine bonds with thymine and guanine with cytosine. In long strands of DNA, replication occurs in many regions simultaneously, forming the sister chromatids required for cell division.
Okay, and that's basically the detail that you need to know for DNA replication. It is an enzyme uncoils and detaches the 2 strands from each other, and then another enzyme comes and simply attaches free nucleotides in the correct order according to the complementary base pairing rule. Now remember, base pairing rule is at the Gold Coast, A to T, G to C. Also C to G and T to A. You have to have it both ways. Okay. But why do we replicate our DNA? It's because the cell is now going to divide. So before you can divide, you need two cells with both perfect sets of DNA. And that is why we do DNA replication. Okay, so here's another video for us.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process
That's the cell's
because this is
life
where
cycle.
it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time.
Just need to be able to
So,
tell the story.
ready for the tricky part? Well, because we tend to count chromosomes by the number
really
of
important.
centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So, 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the stages in order. A is because it's the beginning prophase. And it's going to go away later on, but this is a stage where it's actually still there.
So
The chromosomes are visible.
already
In fact, we say
duplicated
they're
themselves,
condensing,
okay?
which means they are thickening and visible. The next stage is middle because up in the middle of the
Now they
cell.
all go to the
The
middle.
nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there.
They're ready to be
Next,
pulled
the
apart.
A is for anaphase. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last, think chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells on the chromosomes, because you can really, and in the human body, they're each going to have 46 chromosomes. And again, remember,
They have 46
they are
because
identical.
They already duplicated before.
Now,
they-
cytokinesis is responsible for the final
already
separation
duplicated
into
before
two
they
cells
split.
by
Okay.
splitting the cytoplasm,
There we
which,
go.
without understanding
Brand new
cell division,
cell.
because they both require more cells to be made.
Okay, so over here is a diagram on a still image of what you just saw in that previous video by the Amoeba sisters. And what you're seeing is, remember, the first thing that happens is that you have the DNA. Before a cell can divide, it needs to copy its DNA. And that's called DNA replication. And the reason why you need to copy the DNA is because that is all the information that the cell needs to survive. So, if it's going to divide, they both need copies of the how to do it list. And that is what is in your DNA. So, an enzyme comes and unwinds it and then breaks the bonds between the two strands. Then another enzyme comes and attaches free nucleotides onto the nucleotides that is on the different strands, and we call those new strands being formed the daughter strand. But she's always attached at the complementary base pairing rule, so a C will always go to a G, an A to a T to an A. Okay, it's always in the complementary base pairing way. You have to say that, please. And that yellow block there simply is a representation of the enzyme that is creating these new daughter strands for you. And at the end of this, you have the X shape, which is kind of like, let me just make this thicker again. It's kind of like X shape that you always see, and then it's got the centromere here in the middle. But please remember, that is still a single chromosome because it's still attached. We don't say that now it's two. We actually say that it is. I wonder how I make it thicker. Take a highlighter. Oh, that's even worse. But anyways, I hope you understand what I'm saying. And yeah, so now it's got two When you have all of them replicated, they align at the middle line of the cell. The spindle fibers come attached to them, and then they break these two sister chromatids, we call them, apart, and they go to the sides of the cell. A new nucleus forms around it, and the cell can close and form a new two identical daughter cells, both with 46 chromosomes. Okay, so here it is again. The longest part of the cell's life, it's going to be in this phase where you don't see the DNA. It's just doing its thing, making proteins, growing, doing its thing that it's supposed to do. And then when it's become too big or too old, it needs to divide, and it says, okay, DNA, now you condense and we can see them, and then we replicate our DNA. And you can see there, now we have two copies of the same chromosome, but we still call it one chromosome because they're attached. And then they align in the midline of the cell. The spindles come and join to them. It's then pulled apart by the spindles towards the centrioles, and now you can see there that it is now in a separate corner, both of them. And then the new nucleus forms, and then the cell can divide. If you come into the science block again at the bottom floor, with the sliding glass door, it's actually the sliding doors, when it's closed it looks like this, and when it opens it pulls apart the so it's basically mitosis happening, which is kind of cool. Okay, and here's another example, just going through it again. So mitosis creates two identical diploid daughter cells. They have to be identical because they're going to do the same thing, and they need a full set of chromosomes. They can't suddenly have half a set, because they are not egg and sperm that's going to find the opposite gender and then, or the opposite sex, and then fuse to become a new zygote. Okay, these have to be fully functional cells. Alright, so that's where we're going to call it a day for today. Thank you very much for your time and attention. I hope you guys are keeping up, and good luck. I know this lockdown is becoming long, so yeah, just keep on chipping away. Okay guys, have a great day. Speak to you later. Bye.